So I'm going to be talking about the role of lactate in health and disease and lessons learned from elite athletes to apply to clinical populations. And disclosures. So for the past 27 years, I have worked with world-class athletes, professional teams, and international sports organizations in different areas. We're working with, um, you know, from elite athletes, individual sports. I've been working with Olympic medalists and world champions in individual sports, professional sports as well. And it's been a real pleasure for me to be able to work with these kind of populations because it's been a great learning process, and which I still every day I learn from from athletes, from others, and from what I do in for myself and from my mistakes. So measuring the metabolic and physiological responses to exercise has been always an obsession since I was 15. I was reading books, I was testing myself. When I was at the university, I was a guinea pig. And then I started to really be obsessed about measuring metabolism in athletes. Now, I've been focusing on applying this science into healthcare, especially the lessons that we have learned from elite athletes to health and wellness in populations with chronic diseases. Are we still good? I'm sorry, Jerome, can you see me okay? Can you hear me? Okay, great. Awesome. Thank you. So one of the things that has happened in the area and that I've been focusing in the area of metabolism is like we've been in many diseases, we've been going and moving too fast from pathophysiology of diseases to genetics without much understanding of the role of metabolism in many diseases, right? So we know that genetics used to be everything, but at the end of the day, we know it's the science of probability. And at the end of the day, the ultimate metabolic action through all the transcription of the DNA, of the genes into RNA, and then protein, and then a metabolite is ultimately the science of reality. So this is what we're focusing more in our research right away. So that's what we see that the key role of metabolism in many diseases is key. And we're not the only ones, obviously. There are many groups around the world working in this field. And we know that it's key in areas like insulin resistance, metabolic syndrome, cancer, even Alzheimer's disease. At the end of the day, when you see and you deep dive into all these diseases, you see that all roads lead to mitochondria. I'm very curious at the university. I talk to a lot of experts, and more and more of the research, because of the technologies allowing us to understand things better at the cellular level, and all roads lead to mitochondria. And this is why we see that there's so much research coming out right now about the relationship between mitochondrial dysfunction and multiple diseases. And I have these references on purpose here. Some people say, oh, those are all, yes, this is exactly what I want to show, because this is not something new or something that we just come up with this. This is I've been working on for the past 15, 20 years. This is a multiple reference in type 2 diabetes. There are way more than this, obviously, but I just want on purpose show these all ones. Mitochondrial dysfunction, metabolic syndrome, and cardiovascular disease. Then mitochondrial dysfunction is also key in the field of type 2 diabetes. In that I went through that, and there you go. So normally lactate, pyruvate is oxidized in mitochondria, right? And it has to go through its own transporter and be oxidized in the Krebs cycles and electron transport chain to produce ATP. There's always some lactate production, always, which is physiological, and this is the signal. These are the signaling properties of lactate. When it doesn't chronically accumulate, lactate is a very important signaling molecule. I highly recommend this paper that has been recently published by my colleague and mentor George Brooks from the University of California in Berkeley, showing the lactate as a myokine and extrakine, and the multiple benefits for health that lactate has. So lactate can, again, as I mentioned, increase brain function, increase pulmonary function, cardiac output. It's a very important also for generation of ROS, free radical species, which at a high level can be pathological, but at a low level, ROS free radicals are also very important to molecules. It's important for muscle function. One of the things that lactate does is elicits lactylation, which is a acetylation of the histones. Histone acetylation decreases fat metabolism and also activates multiple pathways in skeletal muscle. It also is very important to increase angiogenesis, which is the production of new vessels, and also increase mitochondrial biogenesis as well. During exercise, decrease fatty acids. This is a mistake. It should be up instead of low. It regulates tumorigenesis or carcinogenesis, and also interferes with adipose tissue metabolism. So this is a very interesting paper that has been just published by Brooks, showing the benefits of lactate in health and as a single molecule. My research, and also along with Brooks, has been in the field of the pathogenesis of lactate. So normally, every eat, lactate can be produced from different sources, right? Obviously the source is always glucose. I always say that lactate is the son of glucose, but also the mother of glucose, because it can become also gluconeogenic. But mainly the pathway, like no matter what kind of carbohydrates that we eat, even fructose also, which is not metabolized by skeletal muscle, has to be converted to glucose in the liver. About 80 to 85% of all the glucose in a postprandial state and during resting conditions is metabolized in mitochondria of skeletal muscle through pyruvate. So glucose should be able to enter the cell through the GLUT4 transporters, which are translocated by signaling through insulin, the receptor, and then pyruvate enters mitochondria through its own carrier, a mitochondrial pyruvate carrier, and enters the Krebs cycle for energy purposes. Lactate is always produced, as I mentioned. It's mandatory. Is a mandatory byproduct of glucose, and it's always produced. And lactate also can enter mitochondria through its own transporter, MCT1. Then I'll talk later about CPT1 and CPT2, which are the transporters for fatty acids, mainly for long-chain fatty acids. From glycogen storages, lactate can be produced. So normally during exercise, we have two types of muscles. We have the slow twitch and the fast twitch muscle fibers. And the slow twitch muscle fibers, they mainly prefer fatty acids for energy purposes, which are oxidized in mitochondria. This is why these type of muscle fibers have the highest mitochondrial content, but also they use glucose. So there's always like a little bit of a misinformation around slow twitch muscle fibers just using fatty acids, but they also use glucose. But then as exercise intensity increases, the slow twitch muscle fibers are not fast enough to sustain muscle contraction, and also fatty acids are not fast enough to produce ATP at the rate needed for muscle contraction. So this is where the fast twitch muscle fibers, they kick in and they start to be recruited, and for that glucose becomes a major energy source for muscles. And then finally, we have the very fast twitch muscle fibers, type 2B or type 2X, where not even glucose is enough, and we have to rely on ATP and phosphocreatine systems. So all these are going to be the major lactate producer, fast twitch muscle fibers, because as I said earlier, lactate is a mandatory, obligatory byproduct of glucose metabolism, right? So when this happens, there's an acidification of the muscle microenvironment, not by lactate itself, but the hydrogen ions associated to lactate, which can interfere with muscle contraction, both the force of contraction as well as the speed of contraction. So the way we have humans to oxidize lactate, and this is was a major work by Dr. George Brooks, was what's called the lactate shuttle, where we can shuttle lactate from fast twitch muscle fibers to slow twitch muscle fibers. And this happens in mitochondria of slow twitch muscle fiber. So this is another major source. So if we look at this from a different angle, the glucose is oxidized pyruvate, and then lactate is produced. And lactate has to leave the fast twitch muscle fiber through a transporter, MCT4, and enters the adjacent slow twitch muscle fiber through another transporter, MCT1, and into mitochondria. If we blow up mitochondria, this is what we find: lactate entering and mitochondria through MCT1, and it is oxidized to pyruvate, that which enters acetyl-CoA, enters the Krebs cycle for energy purposes. So any decrease in function of lactate oxidation is going to lead to an increase in accumulation of lactate, which can be pathological. As I'm going to show in the next slides. So again, lactate should enter successfully the Krebs cycle, and also as its own carrier transporter, which is the mitochondrial, the MCT1. So there are different ways by which lactate is it accumulates. One is through the product of the form of exercise. The higher the glucose utilization that the muscle needs, by just mass action mechanism, there's going to be an increase in pyruvate. And there's a lot of pyruvate accumulating in the cytosol that cannot be successfully transported into mitochondria by the carrier, mitochondrial pyruvate carrier. That carrier might be saturated, and therefore that excess production of accumulation of pyruvate is going to be reduced to lactate. This is very important also because during the process of glycolysis, NAD plus is depleted, and the only way to rescue NAD is the reduction of pyruvate to lactate to rescue NAD for the continuation of glycolysis. So therefore, lactate is very important to accomplish this. But when lactate accumulates, we need to oxidize it within the cell. So for that, we have different mechanisms that can alter this lactate metabolism. So one of them is like a decrease in the transporters of lactate, the MCT1s. When you have a reduced transporters of lactate, lactate cannot enter the cell and be oxidized to pyruvate within the mitochondria. Another possibility is a decrease in mitochondrial pyruvate carrier, the transporters of pyruvate into mitochondria. Another one is itself a decrease in mitochondrial function through respiratory capacity and decreases in the whole Krebs cycle. And another possibility is a decrease in the mitochondrial respiration through the electron transport chain. There are different complexes that might not be working properly. So if any of these elements are not working properly, lactate is going to accumulate in the cytosol, leading to pathology. And this is what I'm going to be talking now into different diseases. So the role of lactate in cancer, it was discovered in 1923 by a German physiologist and Nobel laureate Otto Warburg from Germany. And he discovered 1 century ago this year the process of the transformation of the first cell of a normal cell into a cancer cell. So differentiated tissues normally what they use glucose, they oxidize it to pyruvate, and that pyruvate, as I mentioned earlier, is successfully oxidized through a Krebs cycle into ATP. Some of that lactate is always produced, as I mentioned earlier, and it's for signaling, positive signaling um, 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 mechanisms. However, in proliferative tissues, it's been known forever, but now what we discover is that that glucose is not always, that pyruvate is not always oxidized in mitochondria, and a lot of lactate is produced. So this is what struck Warburg the most, not just the high rate of glucose oxidation or utilization that cancer cells have, but the lactate. And this is what he shows in the first English written paper in 1925, where he talks about the metabolism of carcinoma cells, where he showed that when he was adding different substrates to cancer cells, amino acids, fatty acids, and glucose, and he saw that when he added glucose, lactate appeared and showed up, right? And inhibited also cellular respiration. Back in the days, they didn't have millimoles to quantify lactate. They did it in percentage of the weight of the entire cell. So the average tumor, according to Warburg's experiments, was about 12% in weight in the form of lactic acid or lactate per hour, right? So a large amount of lactate, and therefore, and this is what was called the Warburg effect, discovered by Warburg 100 years ago. So again, that excess glucose, that is a typical characteristic of cancer cells, leads to a high accumulation of lactate. This lactate, unlike the lactate from exercise that is cleared by muscle cells or by shuttle, like I mentioned earlier, the lactate shuttle from fast twitch muscle fibers to slow twitch muscle fibers, in cancers, in cancer cells, it accumulates, and that what it dysregulates different mechanisms, as I'm going to show here. But I just want to show how small the world is and how things are related. This is a fun fact. So everything starts with Albert Einstein. So Albert Einstein was very good friends with Warburg's family back in the days of the pre-war in Berlin and the World War II. But at that time, before the World War II, Warburg was already deployed to World War One, and he was fighting in the trenches, and Warburg's mom uh, asked Albert Einstein to please write Warburg to leave the battlefield and come back to the laboratory to continue his experiments. So Einstein was very good friends with the family, and obviously, wrote Warburg, and he was able to bring Warburg to the laboratory outside the trenches, and that's what then Warburg started to continue his experiment. Back in the days, Otto Warburg was the mentor of Otto Meyerhoff, who was along with Emden the discoverer of glycolysis. So both Warburg and Meyerhoff were working on the mechanisms of glucose oxidation glycolysis into pyruvate, without knowing what was happening at the mitochondrial level, but they have a very deep understanding back in the days of how glucose was working. Warburg was also the mentor of Hans Krebs, which, as we all know, is the person who put together all the pieces of the Krebs cycle, right? So all the cellular metabolism that we know from glycolysis to the Krebs cycle came from Warburg's laboratory, and all these three researchers, they always found lactate that was there, whether it's from glycolysis or from the lack of um, um, proper function of Krebs cycles. Lactate was always accumulating until, but back in the days, they didn't have the technology to understand why was this happening until George Brooks from the University of Berkeley in California started to work 50 years ago into the mechanisms that could lead to lactate accumulation. And so from Einstein to Brooks, it's a long journey, right? But he was responsible for bringing work back from the battlefield into the laboratory, and therefore be able to accomplish so many different great discoveries in the area of cellular bioenergetics, including cancer metabolism. So it's a fun story I like to tell. So Warburg was working, but he was stopped by the lack of technology back in the days. And he was trying to fight and try to understand the Warburg effect until in 1953, Watson and Crick discovered DNA and everything changed. So upon the arrival of genetics, everything that Warburg was doing was buried and forgotten until recently. So for decades, um, generations of cancer researchers forgot completely all along about cancer metabolism, and everything was focused on genetics, right? This is the cover of Time magazine saying genetics, the future is now. This was the cover in 1994. But yeah, it's just like um, now Watson's still alive, and he was the father of the Human Genome Project, which led to the Cancer Genome Atlas, the Pan Cancer Atlas. And upon the completion of that, the whole thing was this endeavor was to understand every single mutation that was happening in cancer, and therefore all cancers were going to be understood very well because they were going to be able to understand each gene that was mutated for each cancer. Now, all this brought more answers, more questions than answers, and a lot of anxiety in the area of cancer genetics. And there are multiple papers like this one that says that the end of Cancer Genome Project prompts a rethinking. Cancer needed to be rethought. It prompted a rethink about what was dying, right? What we've been doing, from should shift from sequencing genomes to analyzing function. There's like the highest cited researcher in cancer in history is Bert Wolgenstein from Johns Hopkins, and he had like a very famous paper 10 years ago about dark matter, saying that there's something about the oncogenes that was triggering or affecting the oncogenes. But he called it dark matter because there could be multiple elements that we didn't know. And even Watson today says that targeting metabolism is more promising avenue in cancer research rather than their gene center approaches, right? That locating the genes that cause cancer has been remarkably unhelpful. And if he were to do it again, he would go to biochemistry rather than molecular biology, right? He also says that we still don't know the reasons of the Warburg effect, and that we needed to unleash biochemistry, which was essentially gone because everybody moved into DNA, right? So all this rethinking about cancer, and along with the influential work of Watson, and kind of going back and saying that pretty much we had to rethink the whole um, um, mechanisms of cancer, has led to a renaissance in cancer metabolism. Till the turn of the century, there were no papers citing the Warburg effect. Now we say that at the turn of the century, especially by about 10 years ago, a lot of papers they're starting to cite the Warburg effect, and now pretty much any cancer paper, whether it's in the abstract or in the first paragraph of the introduction, they're already talking about the Warburg effect. Um, so this is where I started to write about um, this because I did my thesis on what we call aerobic glycolysis or cytosolic glycolysis, right? And one night I was looking into, because I heard a lot about the Warburg effect, but I thought it was everything about genetics, and I had no idea. And one night, about 8 years ago, 9 years ago, I started to look into this, and I saw that it was the, Warburg effect was a metabolic feature of cancer, and it was the subject of my doctorate thesis. Uh, um, um, in cancer, this is called a Warburg effect. In exercise metabolism, we call it cytosolic glycolysis or aerobic glycolysis, right? So that's when I started to work together in this hypothesis. And then I took it to my colleague, George Brooks, and then we put it together and we published it. At first, when we were presenting it at conferences, people wanted to throw tomatoes at me because it was a crazy concept. And now, yeah, about 5 years later, has about over 500 citations. So it's been quite well received and has contributed to the spark of the involvement of lactate as a major regulator in cancer. But we couldn't be we weren't be able to do this if it wasn't from the lessons learned from elite athletes in exercise physiology and metabolism, which has been key for us to unveil and be able to try to explain the Warburg effect. Uh, so we in our hypothesis, and I recommend you to read it further if you want to understand a little better of these processes and what we talk about. We didn't discover anything in this hypothesis. We just put together different pieces of the puzzle that they were out there and that they need to be put together to try to explain the purpose of the Warburg effect. So we know that lactate is a master regulator in the major processes in cancer, like angiogenesis. It's key in the immune escape. As we know, cancer cells evade the immune system. It's also key in metastasis and also key in what's called the self-sufficient metabolism. These cells are immortal. They're inefficient, yet not they don't become apoptotic, and they're immortal. So I recommend you to read this in further detail and the multiple steps involved in lactogenesis, which we explained the Warburg effect. And our conclusion or our hypothesis is like lactate is the explanation and the purpose of the Warburg effect, not for substrate utilization purposes or biomass, as some researchers are working now on, but for signaling purposes. And this is what we came up also with what Wolgenstein said, right? About 10 years ago, but there's something about the oncogenes. So that prompted to continue our research further than a hypothesis. Is like, could we prove the lactate is in fact involved, could be part of that dark matter, right? Could lactate be an oncometabolite capable of expressing or regulating the expression of multiple genes involved in cancer? So this is I think someone is speaking in the background. Yeah, so is an oncometabolite capable of regulating the transcriptional activity of key cells key genes involved in breast cancer cells. At the same time, a group from the University of Chicago, led by Zhang, was discovered that lactate it's a it's a epigenetic modificator by histone acetylation, what is called lactylation. So what lactate does, it travels to the nucleus, and over there, it opens up the histones and directly stimulates the transcription of those genes from chromatin. So what we and lactate increases histone acetylation, as I mentioned. But what we discover is like, yeah, we knew that not only it's capable of transcribing the genes, but we identified multiple genes that were transcribed by lactate and either turned on or off by lactate, and many driver genes involved in cancer. We saw that I was like in some genes there was an increase in expression between 150 to 800%, and therefore lactate is a major regulator in carcinogenesis. Then now we have another paper now under review. We've been working in different lines over the years. We have multiple experiments that we're finalizing now that we need to publish. And one of them now is currently under review, where We added an extra cell line, which is a triple negative breast cancer, which is very aggressive. And we added to the regular ER positive, estrogen receptor positive. And we saw, like in this slide, shows that where we incubated these cell lines in glucose media without adding any lactate, just glucose and under fully aerobic conditions. And we saw that both at 6 hours and 40 hours, but especially obviously after 40 hours, there was a very high lactate production, um, especially in triple negative cells, which is more aggressive. We know in cancer that the more lactate that is produced, the more aggressive the cancer is going to be, the type of cancer. And but we see significant increase in lactate without any added lactate. Just it was this lactate is glucose direct lactate, and this is the Warburg effect, where we see that glucose, the pyruvate, is not fully oxidized in mitochondria and is reduced to lactate, and this is what we see here. Then we added lactate as well because we wanted to see what was the addition of extra lactate, like 10 millimoles or 20 millimoles of lactate. But we see that there's not much effect in the total addition of lactate, which has to do very possibly with the transporters, because when lactate is high inside the cells and you add lactate outside the cells, lactate moves from a high gradient to a low gradient. And when the gradient of lactate is higher outside the cell than what is inside the cell, not much lactate is going to be able to get out of the cell. So this is what we could see here, the net lactate release. So, but what we see is that lactate, as Zhang group from Chicago, they discovered lactate travels to the nucleus, and over there it increases the expression of multiple genes. In this case, what we showed is that lactate increases the expression of oncogenes, transcription factors, cell division genes, and is involved in the carcinogenesis through angiogenesis, immune escape, metastasis, and cell-sufficient metabolism. Um, this is under review. So we both see in the two cell lines, there are multiple genes. There are either turned on or even off, like some tumor suppressor factors, like the famous BRCA genes. And also what we did was look at the protein expression. So we saw that in the different experiments, lactate increases the protein expression of multiple genes as well. So we corroborate what we saw in the qPCR looking at the genetic expression as well. We have done the same thing in lung cancer cells, both in non-small cell lung cancer as well as in small cell lung cancer cell, which is very aggressive, and we see very similar results. So we have to publish this data as well. Then some people are saying in some of the critics is like, how do you know that when you incubate cancer cells in glucose, it's not glucose and it's the lactate? So for that, what we did is using genetic engineering, we inhibited LDHA, which is the enzyme that reduces pyruvate to lactate. So by inhibiting here, as we see in the Western blot with the silencer, short harping RNA, there's no lactate production. I mean, there's no LDHA expression in these cells, and therefore there's no lactate production, right? So what we see with this is like when we use the inhibitor, there's in any experiment, and this is specifically to EGFR, which is a major gene that is overexpressed in most cancer cells, and that protein is not even expressed when we inhibit LDHA, therefore lactate. Then we did different experiments where we add lactate when we don't have the inhibitor. EGFR is expressed at 6 hours, and I mean at 24 hours. When we add the inhibitor, EGFR is not expressed, and the same things situation that we see in 48 hours. So with this, we show that it's not glucose what increases the expression of these genes, but it's lactate itself. And we see the same thing in HIF1 alpha, which is a major driver also of carcinogenesis. So we see that, yeah, when we inhibit lactate production, HIF-1 alpha is not expressed both at 24 hours and 48 hours. Um, Again, EGFR is very important. Um, um, uh, the involvement of EGFR is huge in cancer because it leads to the signaling pathways of the RAF, MEK-ERK pathway, as well as especially the AKT-mTOR pathway, which is very important in cancer. So we're working also looking at the role of lactate stimulating these cancer cells, mainly because, I mean, these proteins, these kinases, mainly because the tyrosine kinase changer right now in cancer therapy. They work incredibly well in so many patients and in so many cancers, and it leads to even full remission in so many patients. But eventually, all these receptors and these kinases become resistant to the different medications. So we're trying to understand this why. And when we block lactate all along, we block the expression of the mother of all this pathway, which is EGFR. We also see that it's very important in lactate, the what is called the epithelial to mesenchymal transition. So when cells are more in an epithelial state, they are at a lower risk of migrating and eliciting metastases. And the typical characteristic of cancer cells that they become more mesenchymal. So there are two biomarkers that they're being widely used to see these, which are ecadherin and vimentin. Ecadherin is not present in the triple negative cancer cells, but it is present. And ecadherin maintains epithelial state. So what we see is like when we add lactate, we decrease the ecadherin protein expression. So these cells start losing the epithelial content and become more mesenchymal. And in triple negative cancer cells, which are more aggressive and they have a much higher metastatic capacity, they don't have vimentin, I mean, ecadherin, but they have a lot of vimentin. And this vimentin is significantly increased by lactate stimulation, and therefore increases chances of metastasis. Then we are continuing with different experiments where we inhibit LDHA, and this is some histological um, um, slides here. So we can see here these are MCF7, which are the ER estrogen receptor positive cancer cells. This is the most common cell in breast cancer. So they're proliferating here, these cells. This is where they're converging and fully proliferating. But when we inhibit LDHA and therefore lactate, we see that these cells cannot aggregate, they can't proliferate and eventually die. We see that curves, both the the rate curves, growth curves. We see that these cells, they grow very nicely, and they produce a lot of lactate as well, and they use the glucose, right? But when we add the inhibitor, which is 922, these cells don't grow much over time compared to control, which keeps growing. So we stop within a matter of hours, growth and eventually apoptosis. Then we look at the metabolic reprogramming. So as I mentioned earlier, the phenotypic characteristic of the Warburg effect is that lactate production, right, where pyruvate is not completely oxidized and is reduced to lactate, right? So by inhibiting LDHA in the cells, we reprogram, all right? We call it metabolic reprogramming, but we reprogram these cancer cells back to a normal phenotype, which is the oxidative phenotype where glucose is successfully oxidized to pyruvate, which enters mitochondria. So we can reverse this process of the Warburg effect. But these cancer cells, since they already mutated, don't like it. So eventually they can't thrive, and eventually they can't grow, and they die. This is other cells where this is a full tumor architecture formed in these cancer cells. But when we inhibit lactate in these cancer cells, we see that those cancer cells, they cannot form an architecture, and they're just floating cells around. They cannot get together and form a tumor architecture and eventually die. So then we use a treatment, and we cannot put it here because we have to complete the experiments. But we have a compound that does the same thing also. Without genetic engineering, it reverses the Warburg effect from a control, which is the phenotype of Warburg effect, going back to a oxidative phenotype. And we will see these images in confocal fluorescence microscopy. The blue color are the nucleus of the cells, nuclei, and in red here you see mitochondria. So when we inhibit lactate through this compound that I mentioned earlier, I mean, that I didn't mention earlier, but I talked about earlier, we see that these two things happen. So there's like a destruction of the nuclei, loss of nuclei, or the few nuclei left are aberrant and not functional. And the second event that we see is swelling of mitochondria. This mitochondria eventually burst, and this happens within 24 to 48 hours. So these cells eventually stop growing and die. So this is another approach that we should be in cancer is immunotherapy. So one of the things of why the T cells, immune cells, they cannot kill cancer is because they detect it and they approach to it to kill it, but as it's been approached, cancer cells develop a ligand, a protein called PD-L1 that binds to its counterpart in T cells called PD-1, and this is called the PD-L1 PD-1 binding, and therefore it deactivates T cells. So T cells cannot kill cancer cells. It's kind of like kryptonite to them, right? So 2019 Nobel Prizes, Allison and Hunjo from Japan, they discovered, they started to work into these elements. So they discovered a way to block that PD-L1, PD-1 binding through pembrolizumab, which is one of the first one, PD-L1 inhibitors and immune check inhibitors, which has led to the new field of immunotherapy, which has been very important in cancer therapy. Although it only works about 10 to 20, 10 to 50% of patients, and mainly in liquid tumors, which are like leukemias, as well as in some forms of melanoma. But what we have seen is that lactate significantly upregulates the expression of PD-L1 versus control, and especially over hours incubated, those cells significantly increase the expression of PD-L1. So we're showing that lactate is also a regulator of this protein, and we can block it, and therefore we won't have the expression of PD-L1, and hopefully the T cells can be more effective at, at also killing cancer. We're working also then changing gears, and I'm sorry I'm running a little bit late because of the delay that we had at the beginning. So we're working now on the role of lactate also in different diseases like the pathogenesis of type 2 diabetes and cardiovascular disease. So through muscle biopsies, and we use muscle because, as I said earlier, about 80 to 85% of glucose under postprandial conditions and also under resting conditions has to be disposed in skeletal muscle. Therefore, skeletal muscle is a great organ to study diabetes. So we do multiple assessments with muscle biopsies. This is, we do exosomes, we do proteomics, metabolomics, and but I'm focusing specifically here now and looking in mitochondrial respiration, where we get a muscle biopsy and we enter it, we homogenize mitochondria and we enter it and we inject, like here injecting substrates like fatty acids, amino acids or pyruvate directly into mitochondria and observe what is the mitochondrial respiration and oxidation of substrate. So, and we get a report. So this is what we see that, for example, this is a person with type 2 diabetes. and this is a person that is normally a moderately active individual. So we see that this person has about the people with type 2 diabetes about 3 times lower capacity of oxidation of pyruvate. They have about 2 times lower capacity to oxidize fatty acids, and have about 3 times also lower capacity to oxidize glutamate. So we clearly know that everywhere we look into what's called a different macronutrients like proteins, carbohydrates, and fatty acids, mitochondrial dysfunction is there in these individuals. We also look at the difference between just sedentary individuals, people without type 2 diabetes. They're not clinical populations, they're normal populations, but they already have a downregulation in mitochondrial function, both in fatty acid. This is palmitoylcarnitine, is a long chain fatty acid, pyruvate oxidation, as well as glutamate oxidation that's significantly lower compared to moderately active individuals. So this is important to keep in mind because these populations are not clinical populations. This is what historically has been used in medical research as the control. They're like, quote, the healthy sedentary individuals, but we already see that being sedentary also is causing a downregulation of metabolism at the mitochondrial level, as well as the electron transport chain. So that's going to lead, as I mentioned earlier, to disease and lactate production. Um, so the other thing we see is like the transporter, CPT1s and CPT2s are also significantly downregulated, especially CPT1 in this population. Therefore, their fatty acid oxidation is going to have, it's going to be compromised. Um, and as we see, in fact, that the oxidation of substrate of, in this case, palmitoylcarnitine is also reduced in this population. So um, a typical phenomenon that happens in this situation, and we believe this research that we need to publish also contributes to explain the fat droplet, the intramuscular, intramicellular triglycerides. What we see in population with type 2 diabetes is like there's a fat deposit adjacent to mitochondria that doesn't get oxidized. It accumulates as opposed to athletes or well-trained individuals where this increases and is oxidized for energy purposes. But in this population, type 2 diabetes is not oxidized. And so if we look at the content of this fat droplet, is full of diaglycerols and especially ceramides, which are highly involved in the process of atherosclerosis. Therefore, and this is a big area right now, the relationship between this fat droplet, intramuscular triglycerides, and atherosclerosis because and insulin resistance as well. So what we saw here in our study is already population who are healthy sedentary. They have a significant decrease in capacity in both of the transporter as well as the oxidation of fatty acid, which could lead to accumulation of the fat droplet with a lot of ceramides which are released or could be released to circulation and be part of the atherosclerotic process. So, and in fact, we showed in a different study we published last year that cardiomyocytes when incubated in lactate, lactate inhibits CPT1 and CPT2 activity. So we showed that, yeah, it's going to compromise that accumulation of lactate in the cytosol by not properly being oxidized. Is going to exert effects on the fatty acid metabolism and inhibit the transport of them through CPT1 and CPT2. It increases ROS production and it also disrupts cellular metabolism by decreasing the ATP production as well as the respiration of the cells. So this is why we believe also there could be a connection between cardiovascular disease and type 2 diabetes through a mitochondrial dysfunction, where lactate could be also part of this. And this is a slide that is a little bit complicated, but what we see is that, again, if pyruvate is not properly oxidized in mitochondria for multiple problems, as I mentioned earlier, whether because the lactate transporter is not working properly, or the transport of pyruvate, or overall the entire mitochondrial function is not properly in place, lactate is going to accumulate. So when lactate accumulates in cytosol, um, um, lactate inhibits the mRNA expression of the GLUT4s, which are the transporters of glucose. GLUT4s have to be respond to insulin, and they are translocated to the surface of the cell. And lactate inhibits the expression of those transporters. So right there, you can cause a problem in glucose uptake by the cell. We know that lactate, as we shown, decreases the expression and activity of CPT1 and CPT2. So you're going to have a problem here with fatty acid transport across cells. And it also inhibits, and I didn't show in the previous slide, but we've shown it, cardiolipin, which is a phospholipid that we're going to hear a lot in the next year. I'm sure because it's a major component of both maintenance of mitochondrial function, matrix, structure, and it's highly compromised in with lactate as well. So when we have high levels of lactate that they're not oxidized properly in mitochondria, their signaling properties, they become pathogenic. And this is an area that we see both in pyruvate, in type 2 diabetes, Alzheimer's disease, cardiovascular disease, aging, and cancer. There's a decreased rate of pyruvate oxidation. There's a decrease of glycolysis in these diseases as well, except for cancer, where it's increasing glycolysis to produce lactate. But all these diseases, whether it's type 2 diabetes, Alzheimer's disease, cardiovascular disease, and aging, if we can consider a disease, and cancer, they show increased lactate production in cytosol. So lactate is a major regulator of or dysregulator of multiple events at the cellular level. I just published this study last month, and you can read this in more detail also. So one of the things that I can that I started to years ago, like, can we measure mitochondrial function in a way that is not through muscle biopsies, right? So I developed a methodology to indirectly measure mitochondrial function and metabolic flexibility by measuring fatty acids as well as lactate. So in the laboratory, we use the metabolic cart, where we can use through CO2 and O2, we can through a stoichiometric equations, which have been validated many years ago, we can see fatty acids. And I started using this methodology around 2005 with athletes. But then also I use lactate, which is another surrogate for mitochondrial function. So fatty acids, as I mentioned earlier, can only be oxidized in mitochondria during exercise, as well as lactate. So by combining both fatty acid oxidation as well as lactate, we can have a good understanding of what's the mitochondrial function indirectly of an individual. I presented this at a conference in Berlin, and I published that with George Brooks. And this is our study where we see that this is the blood lactate, and this is a workload, what we increase in power output. These are elite athletes, elite cyclists to the France level. These are moderately active individuals, and these are populations that are metabolic syndrome. It seems that this is deceiving because it seems that moderately active and metabolic syndrome individuals are close together, because this is because they're in comparison to the world-class athletes. So, but we take this population out of the equation, the gap here is significant, right? So, but we started seeing that, yeah, people with metabolic syndrome, they already have very low exercise intensities. They have a poor lactate clearance capacity versus people with elite athletes who have a very high lactate clearance capacity. And that denotes indirectly, it's a proxy for mitochondrial function. Same thing as fat. So world-class athletes have very large amounts of capacity to oxidize fat, huge fat oxidation rates. Moderate active individuals are very good, and this should be what we should have under normal conditions. And then we have metabolic syndrome individuals, which have a very poor capacity to oxidize fat. So I combine then both fat oxidation and lactate clearance capacity, both in elite athletes, moderately active individuals, and individuals with metabolic syndrome. and the correlations are very strong, which shows that this could be a valid method to indirectly um, show measure mitochondrial function. The profiles are different, as you see, because obviously these individuals have a much poorer mitochondrial function than the elite athletes, but the correlations between both are pretty good. Now we're publishing the study correlating this with the muscle biopsies. We have the data, we're writing the manuscript, and there's a very good correlation between the respiration and the substrate utilization of muscle biopsy with this methodology as well. So it makes it more robust without the need of using muscle biopsies to see this in the laboratory. This is kind of a way what it is like when lactate is low, first there's not much glucose oxidation, and also it's a higher fat oxidation as well. Also, we see as lactate is high. We mentioned earlier that it inhibits CPT1 and CPT2 as well as lipolysis, and therefore, when lactate is high, fat oxidation is low. So with this, we can see in a way not only measure indirectly mitochondrial function, but use this even for exercise um, uh, uh, purposes. So we see that if we want an athlete or an individual, for example, to exercise at the fat max and exercise where they burn the highest amount of fat, we can identify it and translate this into either watts or pace or heart rate, right? Whatever the measure we want to and identify zones. We have used this methodology already with COVID patients, with long haulers, or also yeah, people who they didn't have the course of COVID was mild. They never were hospitalized. Half of them, we have 50 subjects here. Half of them were they have comorbidities, and the other half were normal individuals that they would do like regular activities. They were active. If some of them even did some triathlons or marathons at a recreational level, but very healthy individuals. But then they developed long COVID, and then they go to the in this case this is National Jewish Hospital is the top pulmonology hospital in the U.S. is affiliated with us with their university, the School of Medicine. So they go there, and their pulmonary function is completely normal, and the cardiac function is completely normal. Yet these people they're extremely fatigued, tired. Even walking up the stairs is a big task, and they cannot exercise anymore. So we start to try to see what's going on with this methodology. So these are the subjects These are the moderately active individuals. These guys here are the people with metabolic syndrome. And here, look at this. It was really surprising to see. These are the long COVID patients. These ones are the ones with comorbidities, and these ones the ones that were formerly in this category, and now they cannot oxidize lactate properly with long COVID. Then we see fat oxidation. We see the same event. These are the more reactive individuals, metabolic syndrome, and look how bad these individuals do. And yeah, so the healthier individuals without comorbidities, they used to be here and now they're here. We still don't know why, but then in a subsequent study looking at or deploying metabolomics technique, we've been able also to see signatures already at rest without the need of lactate testing or fat oxidation testing, multiple signatures showing that in fact, yes, these patients with long COVID, they have a decreased mitochondrial function. And you can read this paper, and I'm not going to go through all the parameters because I'm running out of time. But the whole purpose too is like, can we use exercise as medicine in metabolic rehabilitation, as I call that concept, metabolic rehabilitation to improve metabolic health. So we know that exercise elicits many changes in the expression of key genes involved in mitochondrial function as well as in health. So can we target mitochondrial function in these genes through exercise? So as Peter Drucker, the father of modern analytics, said, what gets measured gets managed, and this is what we need to measure in as much as we can in at a very high detail as many functions as possible in an athlete or in a person or a cancer patient in order to prescribe exercise correctly, right? And we know that yelling at people is not going to work, and we also know that exercise has to be prescribed individually in order to be successful. We know that many algorithms like the 220 minus your age and similar ones don't work. They're very general. So in the era of precision medicine, we also have to do precision exercise. And in the same manner that we do this with elite athletes, where we really specifically prescribe exercise and very specific different training zones, we can do this with populations with chronic diseases as well and deploy a powerful tool of exercise as a major epigenetic effector and elicit many benefits for health. So this is a classic study which was revolutionary when it came out. These are mice that were transplanted with two lung tumors, and I think it was after 4 months of exercise, no intervention, no medication, no nothing. There was a significant decrease in the size of the tumor and the quantities of lung tumors. And we can see here also the picture and we saw there was a significant reduce in the size and the numbers of tumors. So we saw that exercise, as this group continued on, said it's not just for health, but it can be therapeutics, right? So this is an important aspect that now is taking off, and especially in areas like exercise oncology, it's a big field that is just going to get bigger and bigger and bigger. And how it, we can through exercise prescription, we can alter cancer metabolism. So we know that exercise increases mitochondrial function, decreases glycolysis, increases fat oxidation, and increases lactate clearance capacity. Those are the features of that can counteract lactate cancer, because we can counteract the phenotype of cancer. The mechanisms we don't know yet, but could be due through the exokines or myokines. We know this is from our publication that exosomes are key to for metastasis. Exosomes represent the cell from they come from originally, and they're fully loaded with genetic material as well as proteins and enzymes that can land into a candidate cell, land there and unload all the information and transform a normal cell into a cancer cell. So, but likewise, skeletal muscle. This is from Tarnopolsky group in Canada. Skeletal muscle. They also release a tremendous amount of exosomes or exokines, and could better and healthier exokines from skeletal muscle counteract the exosomes from cancer cells. We don't know, but this is a big area of research now. So just to finish, as some clinical cases using the methodology to look at lactate to indirectly measure mitochondrial function and be able to prescribe exercise. This is a case of a 42-year-old, stage 4 colorectal cancer, metastatic, diagnosed in 2015, 56 rounds of chemotherapy, impressive, and no surgery. And he was an exerciser already, but he was not exercising the right way. He was exercising 4 to 5 days a week, 1 or 3 hours religiously, except for the days that he had chemotherapy. But he was not doing things correctly when it comes to exercise. So this is the results when we first came. This is lactate, and these are fat oxidation. So his fat oxidation was quite poor at low exercise intensities and already accumulated lactate at low intensity. So, and again, this is a healthy, a healthy was a cancer patient. What I mean is like someone who exercise religiously, which is something rare to see in most cancer patients. So, but we saw that his exercise was not well dialed in. So we improved his exercise, and within just 2 months, we saw a significant improvement in fat oxidation and a significant improvement in lactate clearance capacity. So again, using fat oxidation and lactate as surrogates for mitochondrial function, this patient had improved for sure multiple parameter involved in mitochondrial function, and therefore could have a better Exosomes, that's the question. Could this patient now had better exosomes that could counteract the exosomes producing from cancer? So one morning, 2 months later, he called me. He had done his last PET scan, and he said, Inigo, I'm free of cancer. And and he said, like, well, I don't know what's going on. I haven't changed anything. I haven't changed any medication, any routine, anything. Just the only thing is exercise, and no signs of cancer. He was in full remission. So could exercise had been a big player into this? Probably, but in combination also with chemotherapy, right? So Rob was his name. He was remaining full remission for 3 years, and unfortunately, it came back. And when cancer comes back, it comes back in full force, and it's very difficult to beat. And Rob unfortunately passed away. But at least it was for 3 years free of cancer, where he was in a bad state, struggling with chemotherapy and health issues. And at least he had very good 3 years where he could ride his bike, do century rides, and enjoy life for extra 3 years. But areas that we need to improve. Then also using this methodology to improve diabetes or reverse it. So this is a patient that showed up and recently diagnosed with type 2 diabetes. And as we see here, very poor levels of fat oxidation also. So through a specific exercise program, this is my assistant, not the patient itself. And she was a walker. So just walking at a very specific exercise intensity, in this case, heart rate, just by walking, she was able to, in 1 year, to improve her lactic clearance capacity significantly, as well as fat oxidation improve it significantly. So she improved mitochondrial function, and she moved away completely from type 2 diabetes to a normal individual just by exercise. So she was able to revert diabetes. So, and finally, a clinical case with a severe mitochondrial dysfunction and recovery in a severe burn patient. So, a history of a 53-year-old sustained a 37% of total body surface area burned. That's huge and lethal for so many people. But he was able to survive and yeah, and started an exercise program. We saw that for the first months he had absolutely no fat oxidation and very high lactate production. So over time, he was able to, through exercise, to revert this condition and significantly improve little by little his his fat oxidation to have it more to a normal level, and also to decrease his lactate, to improve his lactate clearance capacity. And now this patient it's fully active, very well recovered, and has no signs whatsoever, residues of being in the ICU. So, anyways, that's another uh, clinical case, and just to wrap it up, just showing that using lactate not is just been used forever for with athletes and to help us with training programs with athletes, but it's been helping us to explain or to get to even block and cancer and to block it and even as I mentioned earlier, we block lactate in cancer, we can really stop growing and we can kill those cells just by blocking lactate because this is highly involved in cancer. We can get to explain other diseases like type 2 diabetes, and I haven't even touched Alzheimer's disease, which is a big area of research right now in terms of mitochondrial function and lactate as a major player as well. We can also diagnose people in different conditions beyond type 2 diabetes, like I mentioned earlier with COVID-19 and long haulers, and also prescribe exercise to multiple populations, including cancer survivors or cancer patients or people with type 2 diabetes as well. So the use of lactate it has a lot of potential, and we should be hearing a lot in the next year. So I want to thank everyone and especially my team at the university. And yeah, thank you for your time. And I'm sorry that we had these issues that have delayed my presentation quite a bit. My sincere apologies and thank you, everyone.
Thanks, Inigo. And that was a very fascinating talk, and
Thank
a
you.
lot of info that you passed on. And we managed to overcome the technical issues. We lost a few people who had to move on, but we've still got plenty of attendees in the room. So I'm going to open the floor to any questions. If you guys use the raise hand function, then we'll know which order to ask you to present your question. Inigo, just from my side, I mean, obviously you're in this process now. You're identifying various pathways, and as these things develop, they take time to get from there to maybe having some novel therapeutics.
Mm.
But the one that's available to everybody already is obviously the exercise intervention. And in those cases that you presented, you said you had to optimize their exercise intensity. And I think I probably already know the answer, and that you probably prescribed the zone 2 training. Is that correct? And is that what you would say is the optimal training for people with these chronic diseases?
Yeah, thank you for the question, Jeroen. So yes, I mean, what we see is like when, and this is still we're scratching the surface and trying to understand better. But like in the case of the patient that I showed, he was exercising always at a high exercise intensities, and at that those intensities are glycolytic efforts, which are kind of like the phenotype of cancer, right? So you're not, and this is what we have learned also from athletes. Again, lessons learned from elite athletes, right? We know when elite athletes, it doesn't matter the sport, whether it's a very high intensity sport like swimming or rowing, right? The majority of the training of those athletes are not in the high intensity space, right? They usually tend to train at a lower intensity, but also we see obviously in our sports that we work like cycling or marathon or endurance or triathlon, right? So that's the approach that I start to have in these athletes to try to improve that um, more like oxidative phenotype, right? Through maybe improving mitochondrial function, increasing fat oxidation, and in general, oxidative phosphorylation, which could counteract maybe cancer through specific exercise. And again, the mechanisms could be the exosomes. We suspect that could be part of that. But yeah, the regime that we give to this patient is that. We also know that the more aggressive a cancer is, the more glucose it utilizes, right? And therefore, um, we know that eventually patients run out of glycogen. So if you promote a very high exercise intensity in cancer patients, and not only you might promote glycolytic pathways, but also you are going to decrease glycogen, and which is already deeply low because the more aggressive that cancer is, the more glucose it uses, right? So when you run out of glycogen, then you start to tap into protein, right, through proteolysis for gluconeogenesis, and therefore cachexia, which is a common feature in cancer patients. So it might exacerbate those processes. And so, anyways, this is what we use usually. Yeah, that zone 2 for these patients. Yeah, thank you for the question.
Thanks, Inigo. One of our colleagues had to leave, but he left a question before he went, and he said metformin and other similar medications act at a mitochondrial level, and he said he thinks there's some evidence that it might have an effect on reducing cancer growth. What's your thinking about the use of those types of drugs in terms of cancer prevention in the long term?
Yeah, so this is a great question, and yeah, again, we're scratching the surface in this. My humble take on that is like we know that metformin inhibits complex one in mitochondria, right? So, and we know already that cancer cells are very fragile. They either had a mitochondrial dysfunction or they have an abnormal mitochondrial functioning organelle, right? So if you, but yet they're not apoptotic, right? And this is the typical characteristic. They're not efficient. They don't oxidize glucose, right? Through oxidative phosphorylation, they are incredibly inefficient, metabolically speaking, because they reduce you to lactate and produce a lot less ATP than through mitochondrial respiration. So, but yeah, they're not apoptotic. They don't die. They thrive. They're aberrant and they're not functionally, and that's the nature of cancer. But if you inhibit complex one in mitochondrial function, you might push those cells through through the cliff, and you might elicit apoptosis because yeah, that might be the element that those cells might need to really just die because that's where they might not be functioning all alone. That's a hypothesis that myself and more people are having, but we still need to understand those elements. But yeah, that's a very good question. Yeah, for sure.
Thanks, Inigo. Chris had his hand raised, but I see he's posted his question in the chat now. He says, what does it mean if you see very high lactate concentrations at relatively high power outputs? For example, 18 millimolar lactate if you've done 4 minutes at 6.2 watts a kilo. Is that also related to poor mitochondrial function?
Well, not really. It could be a sign of an excellent glycolytic capacity. Like I personally haven't seen 18 millimoles at 6.2 watts per kilogram, and probably you haven't seen that. At least with the type of athletes that we work, which are right, which are very good at that. They're not as explosive, right? But I would say that someone with 18 millimoles. It's tremendous. Doesn't necessarily mean that there's a poor mitochondrial function, but it means that there's a huge glycolytic capacity. And as I mentioned earlier, the mandatory byproduct of glucose metabolism is lactate. So when you see such a high lactate levels at a maximum effort, that means that that glycolytic capacity, the turbo of that athlete, is incredibly well expressed. So it's a good condition as long as then you have the assistant or I mean the assistance of mitochondrial function of the adjacent type 1 muscle fibers where you shuttle that lactate for oxidation as well. But so this is what we want in athletes, right? The best of both worlds, very high glycolytic capacity, but very high also oxidative capacity.
Thank you. Lila, you've got a question.
Yes, hello. I'm actually from University of Latvia, and I've been very interested in professor's work, and I'm a huge admirer. But I have a question about methodological part of your studies. I've been always a bit curious about fat oxidation zero. Is it at all possible physiologically? And what's the way you determine the fat oxidation? I assume it's from oxygen and CO2 production, but the ways of calculating it differ. So what would you suggest the best way to calculate it? And are you concerned about the zero? I don't think it's physiologically possible. I believe there are always tissues that use fat as a fuel, even in diseased states or in very high exercise intensity.
Okay, yeah, well, thank you for your question, but I'm sorry, I didn't quite understand what you meant, fat oxidation zero. I'm sorry, could you rephrase that, what you mean by that?
Well, you're showing that during exercise, even in the beginning, measurements of type of fuel used during physical exercise, you show in your graphs that fat oxidation is 0 grams per minute.
At high intensities, right?
Even in the beginning, yeah.
No,
But
not
even
necessarily in the beginning. In the beginning, because under resting conditions, like even the skinniest individual has about 100,000 calories worth of fat stored. So this is what is the preferred fuel under resting conditions, and this is what the evolutionary mechanism of humans has led us to use fat as a preferred fuel under resting conditions to preserve glycogen, because we can only store about 400, 500 grams, depends on your size, of glycogen. And out of those, let's say 500 grams, 125 grams alone are needed on a daily basis for the brain, glucose, right? So it's a precious reserve of glycogen. So therefore, under low exercise intensities or resting conditions, we prefer to use fat. However, when we exercise and especially do high intensity exercise, yeah, the nature of the fast twitch muscle fibers, they need a lot of glucose, right? And fat is not fast enough to produce ATP. Therefore, fat is not required anymore, if you will. And as we have shown, the effects of lactate are an endocrine and paracrine effect. Lactate binds to the GRP81 receptors on the surface of adipocytes, inhibiting lipolysis in the first place. Cortisol also and lactate decrease the blood flow to adipose tissue, which is going to even decrease the fat transport through systemically from the adipose tissue. And as we have shown, lactate inhibits CPT1 and CPT2, which is transporter. So it seems that the body has checkpoints in place to inhibit fat oxidation at high intensities, so that we use what we really need. And the way to indirectly calculate it is just like through a stoichiometric equation. So, I used the frame formula, which was developed in 1983, but they were already developed a long time ago by Benedict, over a century ago. But they've been redefined, and there's different ones. So for each amount of carbohydrate that you oxidize, you produce X amount of CO2, and you utilize X amount of oxygen, right? And the same thing for carbohydrates and for fat, right? So you can come up with these calculations to see in grams per minute how much fat you utilize, right? And those are the frame equations that I use based on the stoichiometric equations.
Thank you. If I may add, is it okay?
Yeah, Lila, may I just clarify your question? I think, Inigo, what Lila was asking was, obviously with indirect calorimetry, there's always a potential for error, especially with CO2 being developed from buffering. And that I mean, you yourself have told me to be careful in my own measurements with that specific. So I think, Lila, it's widely acknowledged that there are potentials for error in indirect calorimetry, and the most accurate way would be radionuclide substrate, but that adds a lot of complexity to it. So I think everybody recognizes the limitations of indirect calorimetry, but thank you for asking your question. Just for the sake of time, we've got some additional questions. Quick question from Daniel Lopez. Which is the best kit that you would say for measuring lactate concentrations in the field? And what would be your most appropriate protocol to measure that in regularly active people?
Yeah, that's a good question. So, I mean, now lactate meters, right? They're very popular, and yeah, we use and you use Geron all the time, right? We use these ones. Back in the days, we had these machines with based on electrochemistry, and the gold standard is the Yellow Springs, which is a big machine with a lot of enzymes and membranes and reagents, and a big one and very sensitive, and you couldn't, transport out of the laboratory, and they were like a $10,000 to $15,000 or euro. But now we have these small handheld devices. There are multiple brands. In a similar way, it uses a strip, like kind of like the glucose meters, and they're very accurate. Back in the days, they were not very accurate. People would come to the laboratory, and I would face it right against or compare it against Yellow Springs, and there was not a very good correlation. But they've been improving, and other correlations are great. I would recommend Lactate 2. I mean Lactate Pro 2 and Lactate Plus. Those are the two brands. I'm not affiliated with any brands, but those are the ones that we use, right? Geron all the time in the field, and they tend to be quite accurate. So those are good. And the protocol, I mean, you want to be creative, but one thing always to keep in mind is like you want to do incremental tests, right? To see different responses at different exercise intensities. So you can see what is the response of each individual, and also you can start maybe determining training zones. But also these protocols have to be a little long. So lactate is produced in muscles, and it has to travel from the muscle space into into the blood, right? So that takes time, right? So if you have if you use like a protocols that only last 1 minute or 2 minutes, or even three, the lactate you're measuring now is probably the one from the previous workload, right? So that's why I always choose 5 minutes to even is the lactate that was produced 2 or 3 minutes ago, right? As opposed to the lactate from the previous load. So we are still on the same load. So I think that's important when you face a protocol duration.
And Inigo, I've asked people to cap the questions now, because otherwise, we're in the interest of time, we need to finish up. And I guess you need to go to work because it's early in the morning there. But we've got two more questions. Yuri Smiga said, if someone's training on RPE because they don't have access to a lactate monitor, would you tell them to rather go a little bit easier and make sure they stay in zone 2, or go a little bit harder and closer to zone 3?
Yeah, that's a good question. So I mean, I've been using RPE too, and it usually coincides, right, with what I'm measuring lactate. So normally, if you can keep, I always use the talk test, right? If you can do an exercise and keep like a conversational as we are doing now, we're probably in zone 1, right, or resting conditions of very low exercise. If you cannot keep a conversation at all, you're probably already closer to your threshold or zone 3 or above. But if it's a conversation that is it's hard to keep up, but yet you can talk, right? That usually corresponds to a zone 2. So as old school as it could seem, right? It's an effective way when you compare that with sophisticated methods, whether it's fat and carbohydrate oxidation rates or lactate or both. Yeah, that talk test, yeah, it's quite efficient.
And then the final question, Inigo, thanks, from HG. I'm not sure what their name actually is, but they asked, is there any way to quantify the signal for mitochondrial improvement from an aerobic exercise session in terms of duration and intensity? I can't really understand the question. I don't know if you understand what they're asking.
Yeah, I'm reading. Well, might be, like maybe about like what is the dose and the duration, right? So, and the frequency. So I have three pillars that I've been kind of, by trial and error and learning throughout the years. The first pillar is to identify the intensity, right? There are multiple intensities. I see, and this is my opinion, the zone 2 at least is a higher mitochondrial function, because that's what I see in the laboratory. When people come back, they have the highest fat oxidation and the highest improvements also in lactate clearance capacity. Therefore, it should mean that there's an improvement in mitochondrial function compared to other intensities. And I'm not saying that other intensities don't work, right? But this is the one that I see it works. That being said, also, it's not just about zone 2, because you need glycolytic capacity, which is key for competition, right? So you need to train at higher intensities, obviously. But that's the number one pillar in terms of intensity. The second one is the duration. How long do you have to do this? So it depends on your level, right? But I would say to keep a normal metabolic health, you would need about an hour to an hour and 15 minutes, a minimum, right? To really target. People that train 30 minutes, 20 minutes, it's better than nothing, obviously, right? And if you have an exercise forever, sure, you cannot do an hour right away, but might start by 15, 20 minutes and gradually increasing it. But I see about an hour to an hour and 15 minutes, an hour and a half, up to an hour to an hour and 15 minutes. It's a very good time. And the third one is the frequency. How often do I have to do this per week? So one day is not going to do that. 2 days might maintain. And this is what we learn from athletes, that they deteriorate throughout the season. But if they maintain 2 days a week, usually they can maintain these parameters. But 3 is ideal to get something moving, and 4 is, to me, 4 is optimal. So that frequency of per week.
Thanks, Inigo. And I mean, if you quantify that in terms of your week, you're talking 4 hours of your week, which is really in the greater scheme of things, not that much time to optimize your health and to prevent disease. We should all be doing it. It should be our number one priority and not our last priority. So that wraps up the session, and thank you very much for that great talk, Inigo. I really appreciate you getting up so early and giving us your time and sharing your knowledge and all of your research data with us and answering the questions. And thank you to everyone for staying, for those of you who stayed. And we wish you all the best with your research, Inigo, and with the rest of your day.
Yeah, thank you very much, Jeroen, and I appreciate the invitation, and I really appreciate all the attendance to be here and great questions. And yeah, hopefully it was useful. I appreciate it. Thank you so much.
Right. Thanks, everybody, and we'll see you at the next department seminar. Bye-bye.
Bye-bye. Thank you.